questions?
I was looking for the new module for this week on Canvas and I didn't see it.
Let me take a look. Let's see, let's go ahead and screen share. Whoops, I shouldn't do that. I want to go share computer sound. No, I don't want that. Anyone know what week it is? Is it week 4 or week 5? Yeah, week 4 isn't lit up. Come on. All right, any other questions? Let's turn that on. That was something somebody should have emailed me earlier. I did take a look at the emails on, what is it, yc.edu. I did not finish my emails. And I've got a large number of emails in the Canvas website as well. So I haven't looked at those. Any other questions? All right, well, I sent you an email saying that I changed the schedule. I was thinking about that all term long. And I moved lab 9 down to week 6. And that's going to give you week 7 free to finish your infectious disease report. And it is due on Tuesday of that week, July 14th. And then giving you the rest of the week, as well as July 13th and 14th, to work on your unknowns project. I think you're going to need a lot of time to work on your unknown project. We'll be talking about it next week, the unknown project. Any question about any of that? So this new schedule is up in the Canvas website. And I will point out that especially this term, because of COVID-19, I was really questioning the schedule and if we'll need to change it. I think in the future, I will change week 3 and move one of the labs up. And then we can have lab 8 here and then lab 9 there. Was there a question that somebody was going to ask about? If you notice on the schedule, we're slightly ahead of schedule. That's always a good thing. And I have on the schedule for today, we'd be finishing up chapter 8. We've already finished chapter 8, so I'll move on to chapter 13, but we'll go slowly. And there will not be a change on the quiz. Quiz 3 will only cover chapter 8 and then lab 4. So that should be an easier quiz for you because it'll be over less material. Let's close that down. Any questions? I put in the email that you should be well along on your infectious disease project because, well, this is now week 4, and in week 7, your infectious disease project will be due. And although you've only got one lab in week 6, this is a really long lab. You'll need a lot more time to do lab 9 than any of the other labs we've done so far. And if you have questions about the worksheet, please ask. If you're not in the lab with me, then you can send me a question over an email. And I've asked students when I'm grading the worksheet, the lab worksheets, if you have questions, ask me. And some of you aren't understanding the lab. And the only way you're going to understand the lab is if you ask questions. All right, any questions? If not, let's begin with chapter 13. Am I recording this? Let's see how to get a hold of that.
Yeah, it's recording.
Thank you. I'm only seeing one person today. Why is it so small? Okay, so chapter 13, viruses, viroids, and prions. In most of the lessons, I have the major goals, a rough outline. So both the major goals and the rough outlines. If you want to see all of the goals, take a look at the objectives and you can find that. That should be closed. You can find that in Canvas. And take a look at the study guide. The very top will be the objectives. And then there will be notes. So there's the objectives to chapter 13. So know the terms, know what a capsid is, what an envelope is, what spikes are on a virus. You should know the life cycles, and with bacterial viruses called bacteriophages. There's a lytic life cycle and the lysogenic life cycle. Know what a prophage is, what a provirus is, and this term refers to animal viruses. We'll talk about those as well. Know what the life cycles are, a latent virus, a persistent infection, and there's one other, a budding life cycle. Know what prions are and what viroids are. Understand the various characteristics that viruses have, what their structures are, and how we go about classifying viruses. Understand about viral multiplication, because in a sense, that's what viruses are all about, replicating themselves. And then lastly, understand what prions and viroids are. Any questions about any of them? And you're seeing some viruses on the right. So the only goal of a virus is to replicate. And how does a virus do that? Well, it invades a host cell and then hijacks the cell. So when a virus invades a cell, it usually takes over control of that cell and then uses it to make replications of itself, meaning the virus copies itself in the cell. And then the virus escapes. So that is what a virus does. And let's watch a little video on how it hijacks a cell. Are you hearing this? It's kind of
Yes.
through.
Yes.
Yeah. Phage is a bacterial virus.
In the microscopic world of bacteria, a miniature act of piracy is about to take place. T4 bacteriophages. With their spider-like tail fibers and prism-shaped heads, these viral marauders plan to hijack an unsuspecting bacterium and use it as their own metabolic nursery. This E. coli bacterium has seen its last day. Binding to its surface, the virus injects its precious cargo of genetic material into the cell, commanding it to produce a new batch of T4 bacteriophages. In a final act, the bacterium is forced to self-destruct, releasing hundreds of viruses ready to kill again.
All right, any questions about that? So know that viruses are generally inert when not within their host cell. In fact, they display little or no metabolic activity unless they're in their host cell. So viruses require a living host to multiply. They are obligate intracellular parasites, meaning that they are parasites that have to live within their host cell. They multiply within a living cell, and they use the host cell's enzymes, nucleic acid, amino acids, and all the other molecules of the host cell to replicate. A virus has no mechanism for generating ATP, so they rely on their host cell for obtaining ATP. Viruses have a genome that's either DNA or RNA, and it's never both. And that is what their internal molecule is. So if you look at the inside of a virus, and this virus has one side cut out so you can look inside the virus, its internal molecule is a nucleic acid, and that's the genome of the virus. And the genome is either DNA or RNA. It's never both. And outside of the internal molecule that's the genome, there's a protein coat that surrounds the virus. Let's say in this virus, there's only one protein that's just repeated to make the protein coat. That isn't always the case. Some viruses have multiple proteins in their protein coat. Some viruses are also surrounded by an outer membrane that we call the lipid envelope. And so that would be a circle around the protein coat. Not all viruses have a lipid envelope. Any question about any of that? The host range of a virus is the range of cells that the virus can infect. The host range of a virus is very particular and determined by the specific attachment sites on the cell, and we call those receptors, that the virus can bind to and attach to. And so there are usually ligands on the virus that then bind to the host cell receptor. And those determine the host range of the virus. If a virus were to attach to another cell, and even if it were to put its nucleic acid in the cell, because it's not the host range, the virus would not replicate. So most viruses infect the host cells of only one species, like HIV only infects certain cells of the human body, like T4 and brain cells, and I think macrophages. HIV does not infect all cells of the human body. It is true that HIV can also infect one of the chimpanzees, but still the host range is very narrow for most viruses. That's not always true. As you know, there are some flu viruses that can infect horses and pigs and humans. And there's some other viruses that can infect mice and cats and dogs and horses and humans. So there are some viruses that have a much broader host range than most viruses, but most viruses really only infect some cells of one species. Any question about that? And that's determined by the binding attachment molecules on the virus, and then what they attach to the host cell receptor. And then once they attach, they can then infect the cell. Let's talk a little bit about the sizes of viruses. Let me blow that up. So over here, you're seeing a red blood cell. So you're only seeing a little bit of the red blood cell. This is an E. coli, a little bit larger than the average size of a bacteria. And it's about 100 nanometers long by, excuse me, not 100, 1,000 nanometers long by, am I reading that right? That says 3,000 by 1,000. So I'm going to guess that the 1,000 is this. And that's right, 1,000 is the average diameter of a bacteria. So 1,000 nanometers or 1 micrometer this way, and then 3,000 nanometers this way. The largest virus is longest is about 1,000 nanometers, and that's Ebola, although M13 is close to that size. But if you look at the diameter of this virus, it's very small because it's very short on this side. And so the largest virus is actually megavirus, which is something like 0.4 micrometers or 400 nanometers in diameter. So volume-wise, this is the largest virus, but length-wise, Ebola is the largest virus. And generally, this is the size of most viruses. The smallest range from about 20 nanometers in diameter to 440 nanometers in diameter. The smallest virus that you're probably aware of is poliovirus. at 30 nanometers long. And I think most people have heard of Ebola virus for the longest virus. Any questions about that? That's about 1/10 the size of the average bacteria. When we talk about viruses and talk about their structure, remember that a virion is a single, mature, complete infectious virus particle, meaning having all the parts that a virus needs to replicate. For the simplest viruses, there's only two parts it needs, and that would be the protein coat, shown here, and then its nucleic acid, the internalmost molecule of the virus. When we talk about viruses, they can have different nucleic acids. The nucleic acid can be DNA or RNA. It's never both. The DNA can be double-stranded, and that's normal in a cell. The DNA can be single-stranded, and single-stranded DNA is normal in cells. Whenever your cell is going through transcription, that gene is single-stranded. Whenever the DNA is replicating, it's single-stranded around the area that it is being replicated. Any question about that? Well, some viruses are single-stranded DNA, but others are double-stranded DNA. Some viruses have a genome that is a double-stranded RNA molecule. That is unheard of in a cell. We do not have double-stranded RNA. And then some viruses have a single-stranded genome that is RNA, and that's normal in a cell. When we talk about single-stranded RNA, I need to tell you that some of the viruses have a single-strand RNA molecule that we say is positive strand. That is a strand of RNA that can be read by ribosomes. So they would have AUG for the starting codon, and the ribosome can read all the other codons like it was messenger RNA. But some of the viruses have a negative strand, single strand RNA. And what that is the complementary strand to the positive single strand RNA. So instead of being AUG, it would be UAC. Any question about that? Everyone good on that? The genome can be linear or circular. It depends on the virus. And then some viruses have their genome in several segments. And the influenza virus is one that has its genome in several segments. The genome in several segments should not be new to you because humans have their genome in several segments, and we call those chromosomes. Any question about any of that? So when we're talking about viral structure, I mentioned before, we talk about the protein coat, and that's the capsid. It's the protein coat that surrounds the nucleic acid of the genome. The protein coat is composed of capsomeres, so that would be the individual protein units of the protein coat. Now, when we look at this virus here, it's made up of the same capsomere. So it's just one protein repeating all the times to make up the protein coat. That's not always the case. Like this virus has a couple of proteins. And if you remember the way T4 looked, the head of the virus has different proteins than the tail of the virus. So the different units of the virus we call capsomeres. And in this virus, there's just one capsomere, one protein. Some virus, if they have no envelope, like this virus here, we call it a naked virus. All it has is the genome, which can be RNA or DNA, and then the protein code. And so that's called a naked virus. There are other viruses that have a lipid bilayer around the protein coat, and that's one here. And this we call an enveloped virus. Now, the lipid layer does have proteins and carbohydrates in it, so it's not just phospholipids, okay? It does have proteins and carbohydrates, but the main molecule is actually a phospholipid in this envelope. The envelope is external to the protein coat, and not all viruses have an envelope. Only the enveloped viruses have it. Enveloped viruses tend to have spikes, and the spikes are the outermost red region of this virus here. The spikes are antigens. An antigen is just the outermost molecule of something that our host body recognizes, or the host immune system recognizes as foreign. So the outermost molecule of a spiked virus is the spikes. And so the host tends to recognize these spikes as an antigen. Now for this virus, the outermost molecules would be the protein coat. And so that would be the antigens for this virus. The spikes also tend to serve as binding points, so that they're the parts that bind to the host cell. So if a virus has spikes, besides being the antigens that the host immune system recognizes, these are the parts where the virus binds to its host cell. Any question about any of this? When we talk about general morphology, that is a term referring to the shape of viruses. That was something that was discussed, I think it was in the last lab, but maybe it was 2 labs ago. I'm not sure where I am in my grading. And I was surprised that so many students misunderstood the word morphology when talking about a cell. So morphology is generally the shape. And when we're talking about the bacterial morphology, we're talking about, is it a cocci, or is the cell a bacilli, or is it a spirillum? For viruses, they come in 3 different shapes, and we can classify virus based upon the shape of their capsid. So there are helical viruses, and that's shown very nicely with this virus. Supposedly, Ebola virus is a helical virus, and I certainly don't see the helical structure in this virus. So if you don't see it, that's fine, because I don't see it either. And then there's polyhedral viruses. Those are viruses having many sides. And if you've ever seen any fancy dice that have many sides, the polyhedral viruses look like those dice. They have between something like 6 sides and something like 20 sides, depending on the virus. And then there's a third type of virus, and it's simply called a complex virus. Complex viruses can take on any shape you can imagine. And you're looking at complex viruses here, here, and then here. And the bacteriophage T4, it looks something like the Apollo lunar lander. Okay? Any question about that? We'll talk a little bit more about bacteriophage T4, but as you can see, it has a different shape to it. and it has different proteins. This is the head, and this portion is the tail. And the head is made of a different protein of this part, the sheath of the tail, and that's different proteins than the tail plate. And that's different protein than the tail fibers. And actually, it's this portion of the tail fibers that are used by the virus to attach to its host cells. So only this portion of the tail fibers binds to the host cell. Any question about any of that? All right, this is a better view of T4. Inside the head, you can find the viral nucleic acid in the T-even viruses, and that includes T4, which we'll go ahead and talk about. The T-even all look like this. And then they have a head and a tail and the nucleic acid, which is, in the case of T4, DNA. And it's inside the head of the protein coat. Now, the protein coat is both the head and the tail. The tail is composed of the sheath. We'll talk a little bit more about that part. It has the base plate, which you don't need to know about the pins, but you should know the base plate. And then you should know the tail fibers. Okay, any question about the T-even viruses? They have a distinct shape. What happens when a virus infects a host cell? And can a host respond to a viral infection? When a host cell becomes infected with the virus, that will stimulate the host to produce an immune response and produce antibodies that will bind to the virus. So the antibodies will recognize the virus and bind to it, and the antibodies bind to and recognize the surface proteins of the virus that we call antigens. And generally, antibodies inactivate viruses. And that's one way the host can rid itself of virus. It produces antibodies, which then bind to the virus and inactivate it. Now, some viruses have proteins on the outermost part of the virus that can change or mutate frequently. And therefore, different viral strains of the virus, same species but a different strain, can therefore infect the same individual more than once in the lifetime of that individual. And you should know that about viruses, and that happens with the flu virus. And that's why when you have a flu shot, that flu shot will not last for your lifetime. It will only last for about 3 years. And with each year, the protection the flu shot gives you is less. And after 3 years, the protection it gives you is below the threshold that they say you really need to get another flu shot. In fact, generally they advise people to get a flu shot every year because the flu can change drastically, not often, but every 10 years. The flu changes drastically, like about once every 10 years. And if it changes drastically, the previous flu shot won't give you protection. And generally, after 3 years, the flu is slowly changing. And so they say the protection that the previous flu shot gave you is no longer effective, that it's fallen below the threshold level that they say it's no longer protective. Any question about that? Well, do all viruses do that? No, only a few viruses change so that you have to get a shot or a vaccine every few years. Like polio vaccine was developed, both of them in the 1950s, and they're still being used worldwide, so that the polio virus has not changed, and a vaccine made in the 1950s is still working. Okay, and most viruses, they change very slowly. And so if you get a vaccine, the vaccine lasts a long time, at least 20 years. And if it's a live vaccine, it usually lasts for your lifetime. Any question about any of that? Okay, if you have any questions, please ask. There's a debate going on, and that is, how long will the coronavirus vaccine last? And there's a debate saying that this vaccine may not last more than a year. And the reason why they're saying that is some coronaviruses cause human colds, and the viruses that cause the human colds are similar to the flu virus, and that is that they tend to change over time, and they change rapidly compared to most other viruses. And so there's fear that if they develop a corona vaccine, it may only be good for a year or for a few years. It may not last a long time. And that will be a bad thing for those of us who want a vaccine against the COVID-19, the coronavirus that's currently a pandemic in the world. And we obviously don't know how long the COVID-19 vaccine will last, and we won't know until several months after many people are given the vaccine. So there's really no way to know until they develop one, and then it's tested for several months. Okay? Any question about that? That I thought would bring out a few questions, but if there's no questions, we'll move on. Let's talk about viral taxonomy. Viruses can be grouped in an order larger than families, but generally viruses are not grouped in something larger than families, because when we look at the viruses, they have such a small genome, we're not sure of their evolutionary history. Also, viruses tend to change very rapidly and evolve to their host cells, and their genome is small. So we really don't know the evolutionary history of viruses. And that's why we don't group them in groups much larger than a family. Like I said, some viruses do have an order, but most are just grouped into their families. And none of them are grouped into any category larger than an order, like a phylum. Although you can say that viruses are grouped by the cell that they infect, meaning the kingdom that the virus infects. So we have animal viruses, we have plant viruses, and we have bacterial viruses. And so we can group them into the kingdom that the virus infects. Any question about that? So viruses are grouped into their families based upon the type of nucleic acid they have, the replication strategy, and then the morphology, the shape of the viruses. All viral family names end in the name Viridae, and then we group viruses into genera, and the genus name of a virus ends in virus. And then the genera are broken down into viral species. This is a group of viruses sharing the same genetic information, and they have the same ecological niche, meaning all the viruses of a species infect the same species of animal or whatever species the virus is infecting. And then you will like this part about viral taxonomy. Viruses do not have a Latin name. Viruses have a general common name, which is English. Like the polio virus, that is the common name of the virus, and that's the only name the polio virus has. So viruses have common English names. And apparently all of the early virologists were English speaking. And so all of the virus names have English names. Now, when we talk about the human species, we can break the human species down into races. And we can do that with a viral species. But we don't call them races. We call them subspecies, and you designate that by a number. So there's human herpes virus type 1 and human herpes virus type 2. And so the subspecies is the same species of the virus, but it is given a number. And the subspecies is very important for viruses. And an example of that is the virus species of poliovirus. One of the subspecies of polio causes the polio disease. The other subspecies of the poliovirus is one of the vaccines we use to protect people from getting the polio disease. So that tells you how important the viral subspecies are, that it can have really big effects on the disease caused by the subspecies of the same species of the viral gene species. Okay, any questions about that? Nobody's asking any questions, so I'm going to move on. Are you guys hearing me? Hello?
Yeah, I can hear you fine.
Thank you, Christopher. So let's move a little bit more onto viral taxonomy. How are we doing time-wise? Yeah, I've got time. Like I said, the viral taxonomy, we group viruses into family. I'm going to talk about that with the herpes viruses. So the family of herpes viruses is called herpes viridae. We then group them into their genera, and the genus would be herpes virus simplex virus. So the herpes simplex virus is the genus. The virus then has a species name, and that would be the scientific name would be human herpes virus, but most of the time humans would call it the species herpes simplex. So the common name is actually herpes simplex virus, and the scientific name isn't generally being used, the human herpes virus. The subspecies would be herpes simplex type 1 or human herpes virus type 1. Okay, any question about that? So if you know herpes simplex type 2, it's the same virus species, just a different substrain, and it causes a different disease. For growing bacteriophages, I used to do this. You grow them in the lab, And we're talking about by bacteriophages. These are viruses that infect bacteria, and you just grow them in bacteria. An easy way to grow them is to grow bacteria on a lawn, on a plate, and you just smear the bacteria over the plate so that the bacteria are growing everywhere on the plate. And that's why the plate is white looking. The bacteria are growing in a lawn, meaning you don't see individual colonies on the plate. You're seeing the bacteria grow everywhere. And then you grab that plate before it's grown up, and you smear a culture of viruses on it. Bacteriophages are bacteria viruses. And then you spread those bacteria viruses around. Generally, you either put a ml of a solution that has bacteriophages on it, and then just rotate it around so that ml goes everywhere. Or you could put less, like 1/10 of a ml, and then get a spreader, and then spread that 1/10 of a ml over the plate of cells. And then you incubate that plate, and wherever a virus attaches to a bacteria, the virus will grow, and then it will replicate in that cell, and it will lyse the cell, and then release more bacteriophages, and they will go out to neighboring cells, neighboring bacteria, and infect them. And the process will repeat. And after several generations of growing both the bacteria and the bacteriophages, you'll see a plate looking like this. Where it's white, you have a lawn of bacteria growing all over the plate. And then when you have these plaques, that's where a bacteriophage was that went and lysed the cells. And so wherever you have a plaque there, that's because all of the bacteria cells have been lysed by the bacteriophage. And then you can count the number of plaques on this plate, and that's how many bacterial viruses you had per ml of the solution that you started with. Any question about any of that? And I suppose what you could do is scrape that off and then centrifuge it, and then you could harvest the bacteriophages, meaning the viruses. But that's how you grow them up. You grow them up in cells, and you can even see where the bacteriophages are. Let's go back to viral multiplication and say that the viral genome generally only contains a few genes. So viruses are short and they don't have a very large genome. And all they do is they have the genes required for the virus to replicate. So they'll have the proteins needed by the new virion, like the protein coat, any enzymes that the virus needs. And the virus hijacks the cell and then uses the cell's machinery to replicate the virus. Usually the virus takes over the host cell and then directs the host cell to make more viruses. And the virus will use the host ribosomes to replicate, the host tRNA, the host amino acids, host ATP, and many of the host enzymes. There are some viruses that do, what's the word I'm looking for? They do encode some of the viruses, some of the enzymes that the virus needs, but some viruses use host enzymes. And then probably all viruses use at least some host enzymes. But the point is that some of the viruses use host enzymes. And the basic mechanism for a virus to multiply, it's essentially the same for all viruses, that the virus will multiply in a cell, and how they multiply is essentially the same for all viruses. We're going to be talking about how bacterioviruses multiply. And when we're talking about bacterioviruses, we use the term bacteriophages. Occasionally, people will simply use the word phage or phages. Either pronunciation is correct. I will continuously use the term bacteriophage to make you aware that we mean a virus that infects the bacteria cell. Bacteriophages have two life cycles that they use to replicate. And they are the lytic life cycle and the lysogenic life cycle. And these are the only two life cycles that bacteria viruses have, at least that we know of. In the lytic life cycle, the host cell is infected by the virus, and then the virus replicates and the host cell is lysed. And that's why we call it the lytic life cycle. In the lysogenic life cycle, the host cell, which is a bacteria, is infected, but then the virus does not lyse the host cell. And the lysogenic virus lives in symbiosis with the host cell that it infected. Any question about any of that? Any questions about anything? I think before I talk more about the lytic life cycle and the lysogenic life cycle, I think I'll break, but we do have a few topics right here. Talking about viral multiplication, so let's talk about that. And then I'm going to end the lesson early today, because we're ahead of schedule. So when we're talking about viral multiplication, what do bacteriophages need to replicate? What components do they need to make in order for the bacteria to replicate?
A living bacteria.
They need to replicate what?
You need to start with the genetic code.
Yeah, you need to replicate their genome. What else needs to be replicated at a minimum? You need to replicate their, what do viruses have? They have a genome and?
Proteins.
Say again?
Proteins.
Yeah, they need to replicate the viral proteins. So at a minimum, you need to replicate that. And if we have let me blow this up. If we have a DNA virus, it'll need to replicate DNA. Obviously, if it's not a DNA virus, you got to replicate its viral genome. So that'll be replicated. And then you need to make the viral protein. To do that, if it's a DNA genome, you have to make viral messenger RNA. And so the DNA will be transcribed. And then the viral messenger RNA will be translated to make the viral proteins. And that will be at the minimum the proteins of the protein code of the virus. So we'll call that the capsid proteins. And then those have to get together, the viral replicated DNA with the viral proteins, to make the new progeny virions. So in a minimum, this is what you need to have replicated to make new virions. Okay? Any question about any of that? Oops. So that's what we need to get replicated in a virus. It's a little more complicated. We'll talk about how a RNA virus replicates its genome and how it's also translated. But we'll talk about that later. So I'm going to stop right here. And if there's any questions, please ask them. If not, I will see you in the lab at 2:30.
Okay, thank you.
Okay, thank you. Did somebody ask a question I didn't hear? Anthony, do you have any questions? Also, Joe? All right, if there's no questions, I'm going to end.